Thank you. So I really appreciate Subday giving me 5 minutes to talk about all the new retinal drugs. So I decided to take poetic license and only talk about the next generation of anti-VEGF agents, which that I can do in that period of time. Here are my financial disclosures. The ones in yellow are relevant. So we know that VEGF-A blockade has become the gold standard in retinal diseases, starting with the approval of pegadricase, which blocks VEGF-165, followed by ranibizumab, which blocks all isoforms of VEGF-A. But unfortunately, our clinical results aren't really as good as the clinical trials, and it's largely, as we all know, due to poor treatment. We didn't treat enough. So what can we do to improve durability? And if we look at it from a drug development standpoint, there are many things that can be done. So when we give an intravitreal injection, we give a very high dose. Why? Because all these drugs have short half-lives. So the concentration decays over time and eventually drops below a minimal efficacious concentration where the drug no longer works. So we think of all those different variables, what can we change? If you change the half-life, you're changing the slope of the curve, and that increases durability. If you increase the dose, you're making this curve go up again, that will increase the time that the drug will last. And finally, if you change the binding affinity, this drops the minimal efficacious concentration that's required, because lower amounts of the drug is required to block the target. So the first drug to use this idea was aflibercept, and aflibercept was larger than ranibizumab at a higher clinical dose, and it had a sub-picomolar binding affinity. So if you look at this in a mathematical model of biologic activity, it should last roughly 70 days, and this was borne out in the clinical studies using this drug. Now, no talk about anti-VEGF agents is complete without talking about what's coming next, which is a bunch of biosimilars. These are in clinical trials, and some have actually made phase 3 or had positive phase 3 results. So we have ranibizumab biosimilars, we have aflibercept biosimilars, we even have bevacizumab biosimilars. So we have a lot of biosimilars coming very shortly. Now, this past Monday, the US FDA approved brolucizumab. Brolucizumab is this. So the complementary determining region is the most important part of an antibody. And really the difficulty from a research standpoint is to have the CDR target in the right 3D conformation what we're trying to block, in this case, VEGF. So researchers at the University of Zurich were able to use this fusion protein to support the VEGF-A CDR in such a way that you have the least amount of the antibody required to block the target. So you make a very small drug. So if you look at this, the drug is almost half the size of ranibizumab, and you can put a massive dose into the eye. But interestingly, if you look at the mathematical model for brolucizumab, it actually has a lower activity than aflibercept. Now we know that in the Hawk and Harrier studies it had better drying, and that almost half the patients were able to go 12-week intervals. So obviously there are other things, including retinal penetration, that aren't taken into account with this mathematical model. In China, the Chinese FDA has approved Combricept. Over 500,000 injections of Combricept have been given in China. And this drug has a similar activity in terms of what it blocks as aflibercept. It's also similar in appearance. The difference is the addition of domain 4 of VEGF receptor 2, which improves receptor dimerization, and it improves the binding affinity of this drug. It also lowers the isoelectric point, which improves or prolongs clearance time. In fact, when you compare it to aflibercept, it actually binds all the different VEGF family members with tighter binding than aflibercept. So looking at our table, it's larger in size, it has a better binding affinity than aflibercept. So if you look at that in terms of relative activity, it actually has a longer activity than aflibercept. And this is being tested currently in a worldwide phase 3 study where the drug is being delivered every 12 weeks. Abicavir/pegol also has been discussed just recently. The 2-year results were shown. This actually came from the same, similar laboratories at the University of Zurich. So they were looking at the same thing, and how do you get the CDR to be presented in the appropriate way? They did things a little differently. The scaffolding that was used for the CDR was a design anchor and repeat proteins. These stacked together, almost like Lego blocks, to have a very rigid scaffolding for the CDR. The rigidness of the scaffolding allows this drug to have a very high binding affinity, but just like brolucizumab, it also means it's a very small molecule. Well, the people at Molecular Partners decided to do things a little differently. To prolong the half-life, they fused this molecule onto a polyethylene glycol polymer chain, so-called pegylation, which increases the durability. As shown here now in the mathematical model, you can see it lasts about 110 days, and this was borne out in the Cedar and Sequoia studies where 12-week dosing was similar to monthly ranibizumab. Charlie just talked about KSI 301. This is a polymer with a ranibizumab CDR, and as he mentioned, this drug is very large. So because of the size of this molecule, it actually lasts for a very long time, roughly 4 to 6 months in this model. Now, one of the things we need to understand is our body always tries to find a way around. So if you block VEGF-A, you're going to upregulate VEGF-C. And the reason that may be important is VEGF-C activates receptor 2, which is the pathologic receptor. So maybe this is how patients are escaping from our anti-VEGF-A therapies. OPT-302 is a molecule that blocks VEGF-C and VEGF-D. And you can see from the schematic that would be a pan-VEGF family blockade. This is a VEGF trap molecule that's formed using an Fc fragment and 2 different domains from VEGF receptor 3. The phase 2 study was positive with the study showing significantly better vision combined with ranibizumab versus ranibizumab alone. So we'll have to see in future studies how good this drug actually will be. So I hope I've given you an overview of all the VEGF inhibitors that we have in current clinical studies. God willing, we'll see more of these red ones turn green, which would indicate that we get FDA approval and we'll have a way to decrease treatment burden for all these blind